all the participants who are attending this session. So this is actually 10th monthly series, lecture series on scientists who lived so that humanity lives longer and healthier. So today we will be remembering Dr. or like Professor Donnall Thomas, and he was the pioneer. I'm sure like everybody is aware of it, and he's also known as father of bone marrow transplantation. So he got Nobel Prize in 1990, and the speaker we have today is Dr. Seshu Tyagarajan. It's our pleasure to have Manu here, and she will be discussing on cell and gene therapies, challenges and opportunities. So to start with, I will like to introduce Dr. Seshu Tyagarajan. Dr. Seshu Tyagarajan, she did her master's from Savitri Bhai Phule University. Then she did her PhD from Rutgers University. Presently, she is the Executive Director and Global Head, Late Strategy, CMC Strategy, Cell and Gene Therapies, NTU, Novartis Pharmaceuticals, USA. Dr. Seshu Tyagarajan has an in-depth knowledge of this Carti strategy, and like this is something which is very, you can say, promising in today's time, and lot of research is currently going on this. She has an extensive experience with stem cells and also AI models. She has demonstrated success in developing products from discovery through launch. She is equally well versed in biologics as well as CGT platforms with expertise in all phases of drug development. She has done cross-functional global teams also, like she is part of various cross-functional global teams and has delivered multiple BLAs, SBLAs and INDs. She has navigated complex CMC strategies, successful interactions with FDA, EMA, PMDA, clinical and commercial CGT launches. And she is like thought to be like quintessential leader, and especially while solving problems. And she has a very like meticulous approach while dealing such issues. So she has successfully been engaged with FDA, EMA on complex CMC strategies in CGT space. She has also obtained safe to proceed within 30 days of several CGT INDs. She has organized and prepped commercial manufacturing facilities to launch multiple CGT clinical trials. With this, I welcome Dr. Seshu Tyagarajan. And before we move further, I request Professor Kanan to let us know about KK Biotech, a very great initiative by Kanan sir. So over to you, Kanan sir.
Thank you, Yatindra. Can you move to the next slide?
Yes, sir.
Yeah. So as you can see, the whole aim of KK Biotech is to bridge the gap between academic programs, reality, and employability, which I call as R, for the youth interested in pursuing biotechnology. So, and also, as you are attending today, this is the series on scientists who live so that humanity lives longer and healthier. We have had a whole series, and those of you interested can see the website where we have given all the YouTube which we have uploaded. Next slide. Yeah. So as you can see, we started what is called biotechnology education for students and teachers, what is called BEST Club, and we do this program under the BEST Club, where we have various colleges, universities, etc. can participate. We did one workshop on online internship for a month. It was always followed by theory lectures, followed by 2 hours of demonstration, data analysis, and interpretation. Details can be got on the website. And so we also encourage students, teachers, and postdocs to become member and benefit from our network, which we have spread all over the world. Next slide, please. So this is just a sample. So we did one on, these are the topics which we had done. And, I was very fortunate that people who have been associated with me at some point or other, they came forward and did the whole workshop. So we had a very good, beautiful workshop on LCMS and high throughput screening. How are the new strains of COVID identified? So we had a live demonstration for this, so it was a very interesting one, and they learned a whole lot of things in the last 1 month. Plus they submitted a report on the whole thing. Next slide. Yeah. So, and we pay the teachers who come and do these workshops because it's a lot of effort for them. And we normally prefer a group of 20 to 30 students maximum because we can't handle more, because we also correct their assignments, etc., and help them to learn it better. Next slide. So today's talk, as you know, is going to be given by Dr. Seshu Tyagarajan, and I leave the floor now. Yatindra, kindly welcome Dr. Seshu. Yeah. You can stop sharing the screen, please.
Yes. Yeah. So I welcome Dr. Seshu Tyagarajan to share her ideas and like her experience over these years. So, ma'am, you can start like sharing your screen now.
Okay, sure. Thank you. So I just wanted to say first, thank you, Yatinder, for the nice introduction. And thank you, Professor Kannan, for inviting me to make this presentation today. I'll keep it as a conversation, so feel free to ask questions as you go. The idea is to first give you a very high-level overview of cell and gene therapy field, then talk a little bit on Carti and then challenges, and then move on to CMC project management, which you will see is a artery that runs through the companies to help launch, successful INDs and BLAs. So I just want to keep it fun, keep it conversational. So, so let's see how it goes. And everything that I'm presenting today is born out of my own experience in leading programs at different companies and different types, like in biologics and cell and genes. So that's what I'd say. It may be high level for some of you, but just understand that I'm trying to give you a big overview. So it's very hard to go into detail. Each of these slides can be a 1 hour presentation. I'm trying to give you a whole, 30,000 foot view so that, we get an idea of what it is. Now let me try and share my screen and so we can get started. I think something is happening, and then please let me know if you can see in
Yeah.
a slideshow.
Yeah,
Okay.
Yeah.
Yeah, okay, wonderful. So now, as I said earlier, I won't be able to see you folks, but that's fine. I also want to take a moment to thank all the folks that have joined at different times in the time zone, right? For some it's 6:30 in the morning. That's really a challenge, but thanks a lot. And of course, also folks in India, it's not too middle of the day either. So thank you all for joining, and let's take it from here. So my topic is cell and gene therapy challenges and opportunities. And okay, it's not moving. Okay, so the views I'm expressing here are mine, not of any company, either Novartis or previous companies that I've worked at. So cell and gene therapy, both cell and gene therapy, so cell therapy and gene therapy are overlapping fields of biomedical research and treatment. Both therapies aim to treat, prevent, and potentially cure diseases. And both approaches have the potential not only to alleviate the underlying cause of genetic diseases and acquired diseases, but also they have the potential to cure. And there is evidence now that they have cured diseases. So some therapies are very strictly in the cell area or in the gene area. But there are some, such as Carti, that actually transcend both and are considered as both cell and gene therapies. So these therapies, and we'll talk a little bit as we go forward, these therapies work by altering genes in specific types of cells and inserting them into the body, and then they grow within the body, and now they take over the function that they are supposed to take over. So my next slide is going to give you a little fun facts of the brief history of CAR-T per se. CAR-T is chimeric antigen receptor on the T cells. So the rise of CAR-T cells, cell therapies, I would say, is a very it's been going on for 50 to 60 years. But specifically, if we look at T cell engineering, I think it's around the late 80s when the first chimeric antigen receptor was created. What I have here is just snippets of news from different decades or era. So in the 1980s, T cell engineering had happened. Then in 2000s, there was another breakthrough. But the big breakthrough, so there's a lot of research that was happening in academic institutions. But from an industrial perspective, I would say the biggest breakthrough happened when Novartis signed the deal with University of Pennsylvania for broad-based R&D alliance to advance the novel T-cell therapies. And this happened in 2012. From 2012 to 2017, there was a concerted effort to transfer this technology from UPenn into Novartis, then take it through the different stages of clinical trials. And finally, we come to 2017 when Novartis Carti therapy goes to ODAC, which is the advisory committee of FDA. Because this is the first treatment of its kind ever to be approved, ODAC has to provide its majority approval for the FDA to approve. And so this happens July 12, 2017. And in August and September is when FDA approves Kymriah for pediatric ALL. So this was the first ever CAR-T to be approved in the world. Now, what I have here is a picture and a few news articles that have gone around in the last month saying that it's been a decade since the first two patients were given CAR T cells. And they can say Carl June, who's the father of CAR T, has said, now we can say this is a cure because these two patients have been completely cured. And the reason I put this picture here is this is Doug Olson. I had the privilege of sitting next to him at the ODAC. So I just wanted to share a little personal snippet of all this journey that I have taken through. I've been part of this BLA, the next BLA, and also the ODAC meeting. So this is just some fun facts about CAR-T. Obviously, there's a lot more. The next slide will talk a little bit about what is cell therapy and what is gene therapy. So cell therapy aims to treat the diseases by restoring or altering certain sets of cells or by using the cells to carry a particular therapy through the body. With cell therapy, usually the cells are cultivated or modified outside the body before being injected or infused into the patients. However, there is a little difference. These cells may either originate from the patient, which is then called autologous cells, or from a different donor, in which case it would then be allogenic cells. Some examples for cell therapy that are common are CAR-Ts, NK cells, TILs, iPSCs, etc. Now, when we come to gene therapy aims to treat diseases by replacing, inactivating, or introducing genes into the cells, either inside the body, which would be in vivo, or outside the body, which is ex vivo. And some examples are CRISPR-Cas9, adeno-associated viruses, virus-mediated gene therapy, etc., etc. My specialization is more on cell therapy. So for most of the rest of the talk, we'll be referring to cell therapy or cell and gene therapy. So there are two types, as I mentioned earlier, autologous and allogenic. What do they mean? Autologous, and so these are definitions, folks. They can be applied to CAR-Ts, they can be applied to stem cell transplant, they can be applied to any of the cell types that will be used. So autologous means your own cells, allogenic means somebody else's cells. So there are very specific reasons why companies are doing one over the other. And here are some high-level reasons or differences or comparisons that I've put together. So autologous cell therapies, they usually have a long lead time to manufacture. It can take up to 2 to 4 to 6 weeks to deliver the drug back to the patient. Now, if you look at a cancer patient, sometimes 6 weeks can be a very long time during which the disease can progress very rapidly. So this time is a factor that we have to see how to reduce as we continue to modify these therapies. Similarly, cost-wise, so far, the CAR-T therapies are very manual. The steps are very operator-centric, and therefore the cost is very high. In addition to that, the logistics are extremely complex and making this a very big challenge for global use, but we are still doing it. But regardless to say that the cost then becomes high because we have to do a lot of things in the right way. As I said earlier, immunogenicity, its own cells, so we don't have an issue of immunogenicity. However, when you talk about variability, each patient is a batch. So therefore, the starting material leads to a lot of variability in the manufacturing process. And this is an issue that, is not very easy to take control of. And availability, because we have to depend upon patient cells. So the patient's health can impact the success of the manufacturing process.
Okay, just
Um,
1 minute, just one
yeah.
minute. How, is there enough cells from a patient for transfer, do the whole
Yeah.
process?
Yeah, so it depends upon, so there's multiple, so we, if we look at CAR-T therapies, I think we
Mm,
have come around where we say there are enough cells. Very rarely will it be that we don't have enough cells. Then we have to go back and do a second infusion for the
mm,
patient.
mm.
Sometimes there are enough cells, but they do not grow, and the manufacturing does not get completed because you don't make the dose. In which case, again, based upon the patient's health, we go back and do a reapheresis. So this taking the patient cells is called apheresis. And
But I
yeah,
just wanted to know, how
yeah.
do you take out? I mean, do you release it into the bloodstream? Are they sitting somewhere? You inject some drug to do it, or
Oh, okay. So you're saying so now we are looking at the very first step, the patient's own cells. How do we take them out? So
mm,
you give them, the name is skipping right now, but you actually treat the patients a couple of days before, such that the cells will come out. This is for stem cells and bone marrow cells. They will
yes,
come out into the PBSCs. They come out
okay,
in the peripheral blood.
peripheral
But
blood, yeah. Mm,
here, because we are looking at T cells, I think we don't need to treat the patient. We can take the apheresis and that'll have enough number of T cells. But if you're
okay.
going into specified stem cells or specified cells, you do treat them. And I think it's a 5-day treatment for the patients. It
Mm.
can be pretty painful, but it does allow for the cells to come into the peripheral blood
So
system.
you mean they give something like GCSF or something like that,
Yes,
granulocyte
yes,
colony
yes,
separating factor?
yeah.
And
So the
number
stem
two, how many cells you need? I mean, is it based on per kilogram body weight, or?
Yeah, so the final dose is fixed, which, based upon each drug, there will be a dose that's published already. So we have to look at, okay, so this is a very important question. So now we're talking about very deep into the variability factor. So when you are taking this drug to be filed with the FDA, you need to have certain parameters that are set, right? We cannot change it. This is a GMP manufacturing process.
Mm,
So your initial starting concentration is set such that you have to have a range, but there's a maximum and a minimum range. We need to have that initial seeding density in order
mm,
that the cells will grow and ultimately we'll have a chance at making the dose. And that's
okay,
going to change from company to company based upon
okay,
what process they have and whether they're what they're using in the cell culture process.
okay, okay.
Yeah.
But that means these cells have a good half-life.
Yes. So the way the cells, so this you take the cells and then you freeze them on. So we'll look into that slide.
Okay, okay,
Two
okay.
slides
Sorry,
down.
sorry, sorry.
No,
Yeah,
no, no.
just
That's okay. That's fine. Any other questions? Because this is good, right? It's good to clear the doubts, so that way that the rest of the talk you can understand, or it's easier to follow. So I've explained the autologous, right, which is what is has so far been approved right now in terms of CAR-T therapies. When you come to allogenic, there's a lot of companies that are trying to make allogenic CAR-T to overcome some of the challenges that I just mentioned. So, what are the challenges? Time, right? So, we want the drug to be available for a patient. So, if it is made already and kept, can be made in advance, the treatment is available to the patient on demand, and they don't have to wait these precious 4 to 6 weeks to get their own cells modified. And for the same reason, the cost is generally then reduced. And because it's allogenic, you can take material from a healthy donor, manufacture a multiple set of batches. So manufacturing is scalable, and it's then more, it's more accessible to folks. The big thing here is immunogenicity. And yeah, is there a question? Okay. So, because of this immunogenicity, there is a need to knock out the immunogenic genes in the healthy donor, such that then these can be given to a patients or different kinds of patients. And each company is coming up with their own editing platform, and they all say it's the best platform out there. But allogenic CAR-Ts are still not approved. There's no allogenic CAR-T yet. It's still a few years from getting approval. It has its challenges, but companies are moving towards this. Now, and because for all the reasons that I've been mentioning, it's one batch that can go to multiple, one person that can go to many batches. There's less variability, and then therefore you have better control on the manufacturing. And now let's look at the starting material. Here we use healthy donors as a starting material. Therefore, the success of manufacturing is not as much dependent on the patient's health condition. And because of all the factors I just mentioned, there's a good inventory. It's possible to have a good inventory of pre-manufactured product, and therefore accessibility is there. So these are some of the major differences between autologous and allogenic. And I want to pause here for a minute to see if there's any questions.
So what you are saying is that autologous is only approved. Allogenic is yet to be approved.
It's not allogenic CAR-T is not approved. Now, obviously, if you look at bone marrow transplant, that is allogenic, right? You match a donor, but you have to look at HLA typing and what have you. So yeah, the allogenic CAR-T is yet to be approved. There are companies doing, pivotal trials, phase 2 trials, and there's advances that have been made. Recently, I mean, there's a company called Allogene where FDA put a hold on their material, and then they released the hold. So there's progress that's being made.
Okay. Anubhuti, one of the participant has a question to ask. She has raised her hand. Yeah.
Yeah. So hello. If I understand correctly, the autologous cells and allogenic cells, I see that the allogenic cells are easily accessible and easy to incorporate. So I wanted to know under what circumstances do we choose which therapy is right for which patient. I mean, and if allogenic is so easily accessible and easy to do strategy, this is my question too. Why is it stopped, still not approved by the, what are the reason that it is not being approved?
Correct. So hold on one second. Yeah. When I say easily accessible, it's strictly in comparison with the availability for an autologous CAR-T. So it's in this particular category that this is more accessible versus the other. Now, to answer your question, first of all, as I just mentioned, there is no allogenic CAR-T approved. So we don't have a choice of which one to give. Second is, so therefore, because it's not approved, we don't have a choice, number one. Secondly, when the allogenic CAR-T will get approved, it will be in which disease. So, and then within that disease, which line of treatment. So we may not have the luxury of choosing for at least a decade more. But when, allogenic CAR-T approvals will be like right now, we have from an autologous CAR-T approvals, we have about 5 or 6 drugs in the market. So the doctors have a choice of saying, which one could I give to my patients? There's 3 in DLBCL, and then other multiple myeloma, and all of that are coming. So right now, we don't have the luxury of choice. Maybe a decade from now, we will. And if an allogenic CAR-T and an autologous CAR-T are available for the same line. So that's a very important thing. Is it first line, second line, third line, or wherever it is? So that is going to limit our choices as well. And there's one more thing I wanted to mention. It'll come back later. But did that answer your question, Anubhuti?
Yeah. Thank you so much. And the second question, what are the reasons that it is still not being approved?
Yeah.
I mean,
Yeah. Okay. So all of this, right? So this we will look into, but it's good that we are preempting the discussion. So the drug discovery and the drug approval process is a very long process. It depends upon, number one, you need to have a candidate that can go into clinical trial. So that's the very first requirement. But after that, it is what is the trial design? How did you design the trial? What was the endpoint? And then how do you get approval for that particular drug in this particular indication? And what does your label say? Each one of these is a huge topic for discussion because anything that's not done right is going to impact what is called the final label of your drug. So the reason allogenic CAR-Ts are not yet approved is right now because you don't have the right candidate in the phase 1 trials. So people are now moving. First, it was CAR-T. Can we get a CAR-T approved or not? So that hurdle was what should you say, was overcome 2017, 2018, 2019. Now that we have overcome that for the first time, now we are looking at what are the problems with the approved CAR-T. There are multiple problems, and each group is going to and try to address it in different ways. One of them being allogenic. So one of the biggest problem right now with allogenic is the durability of response. Whereas CAR-T, at least we have a few patients that are cured for 10 years. Allogenic so far has not shown that it is a durable response, even in the short term that it has been shown to be good. So we have few years to work on these to say they can be sustainably available in a consistent manner for the population.
There is another person, Ritika Gupta, who has a question to ask.
Sure. Go ahead.
Dr. Seshu, hi. So I have one question. Now, when we
Yes.
talk about the manufacturing process of autologous versus allogenic,
Yes.
how different would they be, and where are the key differences? At what steps?
So I think because there's not much information known about the allogenic manufacturing processes, but what little that I do know, the manufacturing processes are generally similar. So you will take you will take the T cells, whether from the patient or from the another donor. You will modify it. The difference will be in this modification. In an autologous situation, what we are trying to do is we are just introducing a particular gene of interest, which is the CAR-T here, into the T cells of the patient or the own patient, and then we are letting the cells grow in a lab, and then making sure that we can release it as per our quality systems, freeze it, release it, and then ship it. In the allogenic CAR-T, it's still very similar, except that in the gene modification, now we have to knock off the immunogenicity causing genes. And as I said before, each company has a different way. They either use CRISPR or some other, different softwares or different modalities to do the gene editing. And then they have to then again proliferate the cells, which is growing the cells in the lab. And then what happens is in the autologous case, you have one patient, one bag, or one patient, 3 bags, but it's a batch. In the allogenic, it will be one patient, but you can get multiple batches or multiple doses from the same person. But in order to get the multiple doses, what will then happen is you have to grow the cells much longer as opposed to the autologous one. And in that growth is where I believe the manufacturing media is going to be different. I don't know the details because it's not out yet, but my thought process would be, if you're familiar with perfusion versus batch fermentation, this would be more like, how do we supplement the cells to grow such that they are still in a healthy state, they can still produce the protein of interest, but they are not, going into the dead state. So that is where the difference is. But overall concept as of today is very similar between autologous and allogenic.
Understood. Thank you so much.
You're welcome. Any other questions?
No, you can start with the next slide, yeah.
I have at times been 90 minutes on one slide, so I'm perfectly fine with answering the questions. No problem. Yeah. All right. So the next one is, just an overview of the CAR-T receptor. And to just give you an idea of where's the first generation CAR-T, second generation CAR-T, and now we are talking about third generation CAR-Ts. So CAR-Ts are hybrid proteins consisting of a single chain fragment of the scFv fused to a co-stimulatory domain, which is a CD28 or 41BB. Both of these are now in the market, and with a CD3 zeta to mediate the T cell activation. So this is the moiety that is introduced into the T cells of the patient, which then grow, and then we have to measure that this is active That is done by potency tests. And then once we have the desired dose, we will freeze the cells, and then they'll be infused into the patients. We'll look into that. But then what happens is when this T cell that's expressing this receptor is within the body of the patient, they actually proliferate by recognizing the B cells that have the CD19. So that attachment or recognition of the tumor actually proliferates the cells as well. And so they continue to look for CD19 expressing B cells and kill them, kill them. And then that's how the cure is. That's how they cure the cancer or the patients of the cancer. So at every slide, wherever I've taken information that is not mine, I've given a reference here, folks, just so that I'm citing the right references. Now below, I've just given the names of the commercially approved Cartis as of today. Kymriah was the first, as I mentioned. This is Novartis's Carti. This was first approved in pediatric ALL, then in DLBCL. This is diffuse large B-cell lymphoma, and then follicular lymphoma. And just as a side note, I have worked with all of these BLAs. With follicular lymphoma, I set up the clinical trial, and then I moved on to the next generation platform, and then it got approved last year. From Gilead Kite perspective, Yescarta, it's approved in DLBCL and follicular lymphoma. And Tecartus is again a Gilead Kite. It's approved in mantle cell lymphoma. Now we come to BMS, which is basically Celgene and BMS. And Celgene had partnered their drugs either with Bluebird or with Juno. So Breyanzi is the BMS Celgene drug for DLBCL and follicular. Abecma is the BMS Celgene Bluebird drug that is in multiple myeloma. There's the next one that should be approved anytime, and this is Legend Biotech from China in collaboration with Johnson & Johnson. It doesn't have a commercial name yet, but it's called Cilta-cel, and this is awaiting approval for multiple myeloma. And this is the success rates and the remission rates are very high. So this is a very good drug in multiple myeloma. So this is just an overall, where we are with respect to CAR-Ts. The next slide. Okay, so this is our autologous CAR-T, autologous cell therapy. So from here on, I think most of the things I'm mentioning are with reference to autologous cell therapy. And I want to just see if the animation is working. Is it working?
Yes.
Okay,
Yeah,
wonderful. So our first step here is the apheresis or the leukapheresis. We've got the patient here, and in order for the patient to be agreeing or to be acceptable for apheresis, there are a few preconditions. If it's a clinical trial, then they have to have the inclusion-exclusion criteria, and they need to be part of the inclusion criteria. And then there's other tests that they have to undergo before they can be ready for apheresis. And on the date that the apheresis is set, or a couple of days before or after, their health should be good that they can undergo this procedure. All these things are very important. That's why I'm explaining why it takes longer, because there's a lot of things that's not in our control here. So the first step is assuming all that has passed through. First step is apheresis of the patient. This happens at the APH site or a hospital or an infusion center. But this is the clinical site. So here, the patient's blood is drawn through the apheresis machine, and then based upon the criteria for the drug and the treatment, it is frozen. It's collected and then either frozen or it kept at 4 degrees. It's chilled. So for Kymriah, we freeze this apheresis, and then it is shipped to the manufacturing site. For the Kite products, the Kite Gilead products, they bring it in fresh. Now, there's challenges between if you bring it fresh versus cryopreserved. There's both pros and cons for both. That is a topic for another day. I don't think we can get into that. So if we look at the Novartis process, it is cryopreserved, which means it is frozen at the clinical site, and then it is shipped to the manufacturing site. Now, because it is frozen, it can be shipped to the manufacturing site, and we have flexibility in figuring out when we can slot the patient into the manufacturing schedule. That's the biggest advantage of freezing, and also showing that your manufacturing process works on frozen material. This is a very important step. A lot of processes, manufacturing processes, do not work on fresh material, or they do not work on frozen material. So it's good to be able to establish that we can use the frozen material and still show good manufacturing success. So what does the process look like? The first step is called enrichment and activation. And this is done via 3CD28 magnetic beads, Dyna beads. These cells are mixed with the dynabeads, and in the process the dynabeads not only select the T cells, but in the selection process they're also activating the T cell. As the T cells activate, now available transduction. So now, which has the place over 2 hours. There's one dose of vector that's given one, another dose 24 hours later. Then the vector, they are flash manner. So this everything the cells are in days, they move static to dynamic, and then the T cell action happens, and they're growing in a wave vector. Now this T cell expansion can for 5 to 6 days, at the end of which your idea is to make sure you have enough dose in the cells that are available. So this whole process from activation to harvest is about 9 to 10 days, and it is variable. It can be 9, 10. Originally, we also had 11 days, but then finally, when we got the process approved, it's 9 or 10 days. And it all depends upon cell growth. So at the end, assuming that we've got the dose, we've got the cells and the activation and transduction, then the cells are washed, they're formulated and cryopreserved again. And now this is ready. So it will be in the freezer while quality tests are taking place, where we want to make sure it's sterile, it's safe, it's efficacious, it's met all the critical quality attributes. Then our quality system can release the batch. Until that time, this stays within Novartis control. Once the batch is released, it's going the other way. Once the batch is released, now it's sent to the patient, to the clinical site again. Here, when the patient and the physician know that the drug is coming their way, the first thing they do is they give a chemotherapy to the patient. And then sometimes they may give a bridging therapy to the patient as well. Now the patient is ready to take the drug, and now the infusion has happened and the cycle is complete. So this is the total, these are the major steps in the autologous cell therapy. A big element here is the step 1 and step 6. This is totally dependent upon the patient's health at that particular point in time. And it is at the physician's discretion to see when they can infuse the drug into the patient. So I'm going to pause here because it's a lot of information that I shared, and I'm sure there'll be a lot of questions here.
Ritika, you raised your question.
Yes. So I have one question here. So now when we're talking about transduction, so basically what percentage of the cells get transduced? And typically when it's being administered to the patient, what is the percentage that is required to basically give it to a patient? Like, is it 20%, 30%, or how does that work?
Good. Very good question. So there is for every step here, you've got your critical quality attributes and you've set the limits. For some, the limit is greater than, it's a one-sided limit. For some, it's a two-sided limit. For some qualities, attributes, it will be a two-sided limit. For transduction per se, the limit is greater than, and this is all published, so I can share this information, otherwise I wouldn't be. The minimum is 3%, okay? So you need to have 3% 3% of the cells transduced in order for us to show that when ultimately you've got the dose, it will work in the patients. Typically, the target is 20%, and we reach around 10 to 15%. But this again is a lot of variable factors. It depends upon the particular batch of vector that you get from we get it from Oxford. So each batch of vector has its own titer. And then again we do the MOI within the cell therapy area to see how much volume do we need to add. So but there's a batch to batch vector variability in terms of how much volume will be added. So if you have a minimum of 3%, you know that you can make the dose. Then at the other end, you don't want it greater than 20%. You don't want 40, 50, 100% of the cells transduced either, because we have a restriction on the number of gene copies per cell. So we have to maintain that. And in order to maintain that, you need to be within this range of 3 to 20%. Did that answer your question?
Understood, understood. So now when we're talking about 3%,
Yeah.
is this standard for all the therapies, or like does this change from therapy to therapy, company to company?
No, yeah. So I would not say therapy to therapy, but company to company. And primarily, it's your cell culture media which has a big impact on the transduction. If you change the serum, do you have serum-free process? Do you have a serum process? So these things can impact the level of transduction. And then also the dose, right? So what is the dose that each company is aiming towards will depend upon, will then determine what is your percent transduction.
Understood, understood. Thank you.
Mm-hmm.
However, when you say like the 3% is the population which is transduced, so
Yeah.
that 3% is in multiple copies or like single copies only of that particular gene?
A good question. So that now we're going into analytics. I know we have a restriction of less than 2 copies per gene, but beyond that, I cannot. That's what I know.
But again, you also mentioned like you said like you cannot go beyond 40 or 60. So I thought like is it because that the 3% is having multiple copies and that is sufficient to give the desired result actually?
Correct. So because we don't, from an FDA perspective, right? Safety, not only FDA, but from a company perspective, safety is also very important. So that's why you don't want all the cells to be transfused either. And the reality is somewhere in between. Now, do we want to be at 3? No. We want to be in the 10 to 15% transduction. But we've shown through clinical trials that below 3%, then either the dose is not made and the patient may not show benefit of infusing a material that is less than 3% transduced. And then we have to understand that a patient, like giving a material that's not as efficacious, is also a risk to the patient, right? They go through the CRS, they go through a lot when they take this. So we want to make sure that the patient is getting the best benefit.
And ma'am, how does like this lentiviral vectors fare when you compare with adenoviral? Because somehow adenoviral vectors are preferred as compared to lentiviral. But I am seeing here like more of lentiviral vector actually.
That's a great question. I think we may need to have a group discussion. Lentiviral will help with the T-cell transduction, right? Whether AAV will help with that or not, that somebody else may have to jump in.
Okay.
Yeah. And these are all, just by the way, these are 3rd generation replication incompetent lentiviruses. Yeah. Yeah. And we have to show that batch after batch. And especially for the ODAC and stuff, FDA, we had to show that these will not replicate. I think every new batch we have to show also, or at least every 6 months or something, we need to keep showing this.
Okay.
Yeah. Okay. All right. Let's, I'll you can come back and ask questions, but is there any other question on this? So I'm giving a simplistic overview of cell therapy manufacturing here. This does not cover all the elements, but just to give you in blocks, what are some of the terms that we use, so that if you come across these, at least you have some introduction. So the first line here is called make, test, release, which is basically what I just talked in the earlier slide, basically. So what is make, test, release from a manufacturing perspective? There's incoming material, which I said is apheresis. This comes in, we process it, we test it, we've frozen the final product, and now the testing results in releasing, and then we ship and infuse. This is what I just walked through in the previous slide. Excuse me. But what are the things we need to consider in all of this? One thing is consistency. That means we need to show that it makes the dose and the dose is efficacious. So this is a big, I mean, everything that I've put here is a challenge. That's the reason why they have made to this slide. So you need to make sure as you are manufacturing the batch, you're testing it. The potency test shows that this is active. The cells have to be viable. There's a lot of times the cell growth is not great and the viability becomes an issue. We have to be able to make the cell dose. It has to be safe. That means no contamination. Efficacy is a combination of the dose, potency, and viability. It has to be pure. Your residuals need to be less than the required percentage. Of course, identity is important. We have to show that the drug is stable for the period of time that we have said that it is good, or the apheresis is stable. So the stability is either how long can I store an incoming AF material frozen, or how long is the drug good for after I have manufactured. And we can discuss that if we have time. So stability and shelf life go hand in hand, storage, and then specifications. So these are some of the things that the manufacturing group has to overcome on a daily basis or on a weekly basis while they are manufacturing, such that they're able to consistently manufacture a product, release, ship, and the patient is benefiting from what we have manufactured. Some of the other terms that are very common now, if you look at the literature, is called vein to vein, vein to door, door to door. What is vein to vein? Vein to vein means from the patient's apheresis, which is a vein, to when it gets infused, the other vein, right? But and so there's a lot of focus today on tracking the vein to vein time. So from the time apheresis happened till the time the patient has been infused can be in the order of months at time. Even though it may take only 2 to 4 weeks to manufacture the drug, this vein to vein time is very long. So there's a lot of emphasis on reducing the vein to vein time. So what happens in the vein to vein time? What are the things we need to look at? The logistics are a nightmare. So we need to make sure that the logistics is happening in a very smooth manner such that everything is going as planned. So within the logistics, we've got scheduling, we've got tracking of the shipper. Chain of identity is another huge term, and then shipping globally. So these are just, as I said, some things that came to my mind. So I put it here such that it triggers something when you read or when you're talking. It says, oh, I've heard this term somewhere. Now, all this is happening under GMP manufacturing. So therefore, quality systems have to be met, obviously. And if you are manufacturing in a GMP environment, then you have audits, you have SOPs, you've got inspections, you've got oversights, you've got investigations, and then you have to figure out like how to overcome, how to solve the investigations. And now because you've got procedures for everything, if you have to deviate for some reason, then it's a pre-planned variation. So all these things add to the burden of GMP manufacturing, but also add to the number of people that is needed in order to release a batch, adds to the time that it takes in order to release a batch. Any questions on this one? Okay. So, sorry, was there a question?
I can wait, actually. I'm fine, yeah.
Okay, yeah, all right. So now we're looking at autologous cell and gene therapy. This is just another, what are the challenges, right? So these are complex molecules, as you can see. They're difficult to characterize, very fragile, and they can be immunogenic. This is a developing field, so not enough information is available. Why do I say it's a developing field? Because if you look at monoclonal antibodies, there's been work going on for 40 years. So then what happens is you have the whole supporting industry that has also grown the field, and you whereas in That's the point. We are missing if no this by and compare a small molecule or a biologic or an antibody. So I'm just trying to show where cell and gene differs from what is out there right now. So as a result of all of this, the CMC package is highly complex. It is time consuming, and generation of the data can be a pain, and it is a pain. So that's definitely there. I've already mentioned that one batch is one patient and one dose. This is in the autologous cell therapy, so it's adds to a lot of variability. So if you look at defining a manufacturing process to begin with, you need it's a challenge to say, here's my process, this is how I'm going to define it, these are the control parameters, and this is how my process will work time after time, month after month, year after year. So this is in biologics. It's a challenge, even in biologics. Now to this, you're adding, we have no idea the variability of the starting material. So the patient brings in the variability. And we still have to now show that this is under control. Otherwise, FDA will not approve it, and we cannot release it on a day-by-day basis. So this very high variability in the process because of the donor is a very big element to overcome. And sometimes you cannot overcome because it depends upon the patient population. So these are just some of the high-level things that come to my mind. What are the other challenges? Cost of goods, as I mentioned, is very high. It's called COGS. We need highly skilled personnel. So this, as I mentioned earlier, is a very operator-driven process. Also, analytics is very important. It's a growing field. So right now, there's a dearth of technically skilled people who can do this, for multiple reasons. There's hundreds of trials in cell and gene therapy, just in Carti. I think thousands of trials in cell and gene therapy. So there's multiple companies, multiple groups, multiple academic centers that are coming up with trials. All of them need this skilled, trained personnel, and you cannot find enough of them in America for sure, and I'm sure it's the same case other places as well. Next challenge, concurrent development. So we are learning as we are going. So a common term we used to use with Kymriah is that you're fixing the plane while you're flying it because you don't know what's coming next and you're just figuring out and going with it. A lot of these cell and gene therapy drugs are academic to industry transfers. Now, why is this a challenge? Academic institutions have one way of running GMP. Industry has a very high standard of doing their GMP manufacturing. Not only that, FDA has different expectations from an academic program versus an industry program. They hold the industry to a much higher standard. And to top it all, industry has already been, so I'll take industry, meaning Novartis, Merck, BMS. We've already been manufacturing small molecules to a very high quality standard. We have manufactured monoclonal antibodies to a very high standard. Now we come to cell and gene and we say, oh, we cannot say that those standards do not apply, right, as an industry. So therefore, this academic to industry transfers is a big challenge because we have to raise the academic GMP level or the package to an industry GMP level. And I've done this, and I can tell you that it's not an easy. It's a pretty big challenge to overcome. And so the tech transfer challenges, comparability, again, are part of this, saying how do I tech transfer from an academic institution to a company or an industry, and then show that these processes are still comparable. This is again a huge topic. Because of the growth in the clinical trials and what have you, there are not enough manufacturing facilities available in the world to manufacture these clinical products. So access to CMOs is difficult. If you're lucky to get a CMO, then you get like 2 slots a month or 3 slots a month, and you have to by all means make sure you meet that slot, because if you don't meet that slot, then the next slot may be 2-3 months down. So access to CMOs is difficult. assays, as I said, the characterization assays, the potency assays, flow assays, the assay development field needs a lot more progress and a lot more improvements and a lot more people, and we don't have enough over here. Scaling out, as I said before, I think I mentioned very gently, autologous therapies are not good in scaling out, because the scaling out means you're just expanding the facility to make thousands of the same thing. Whereas in an allogenic, you can at least scale out because you have one batch that can be given to multiple patients. As we are going through the challenges, process validation. A successful process validation depends upon rigor of your manufacturing, robustness of manufacturing, yada, yada, yada. Now, if we don't have enough data, we don't have enough material to run as many design of experiments as we want, then the validation has to be very creative, and we have to make do with less information and still show this is the process, here are our parameters, and this is how we're going to define this commercial process, and FDA, do you approve or do you not approve? I already mentioned logistics and supply chain. It's another big challenge. Aggressive timelines. Timelines are always aggressive, right? And I'll walk through, I think, towards the end. But it's always okay. How fast do we get to clinic, phase 1? How fast do we get to phase 2? How fast do we now get into pivotal trial and register? So you're always competing against timelines, no matter where you are in this field. That's for sure. And then, as I said, not enough data. Not enough data because you don't have the luxury to do as many experiments as you wanted before the clinical trial started. The clinical trial size is small. So even from a clinical trial perspective, you don't have enough data. And where would this, so when I talk about AI models and stuff, these are very nascent trials with little patient numbers, such that AI models need a lot bigger database in order to be successful. So these are some of the challenges in this field that I see. The next 2 slides talk about the drug development process, and then we move into CMC project management. So I want to pause here to see if you have questions. Okay, that's good. So from a drug development process, we've got the preclinical, phase 1, phase 2/pivotal, or phase 3 and pivotal, and then we go into BLA and launch. Now, each of these fields or each of these phases has different things that a company needs to do or a group needs to do in order to move from one phase to the other. But high level, preclinical is where you're trying to do a target identification to find which is the one that is going to the next phase, which will be a clinical candidate. So you have you've got a lead, you've got a recommendation, you have an early process and analytics to go with the program, some safety, preclinical, and then IND filing, IND enabling runs. Now you're in phase 1. The main purpose is to show that the dose is safe, and that we are ready to do the dose escalation. Then when it comes to phase 2, the idea is to show that it's not only safe, but also efficacious at multiple doses. We can also do dose finding in phase 1 and or phase 2. I'm not going through all these elements because that's a lot, but we'll just move through quickly. Now, from a pivotal perspective, it's a big...
There is a question from Surya Narayan Rao, sir.
Okay,
maybe
all right.
Take that.
Well, good morning, Dr. Seshu. My name is K.S. Rao. I'm a toxicologist.
Yeah.
So I know you skipped over preclinical safety and IND.
Yeah.
It's my understanding that for autologous cells, you don't need any animal testing, because I remember there used to be a company called Dendrion.
Yeah.
I'm sure you know it. It has gone through a lot of problems.
No, sorry. 1 second. Our facility was or is in the Dendrion facility. So we not only know, we work there. Yeah, go ahead.
Very good. Okay.
Yeah.
In the autologous cells, you don't need any animal safety. Is that correct?
No, I think animal safety, meaning are you talking about monkeys and stuff?
No, I'm talking about the repeat. Normally, if you take either a large or small molecule, you keep on giving repeated dose to see any systemic toxicity in animals, rats, mice, and dogs. Yeah.
We do mouse, right? So that's why I had to take. We do mouse. We look at mouse models before we can move forward.
Okay. All right. Thank you.
Yeah.
Thank you.
No, you mentioned and facility Dendrion, and then there were 100 people that used to work at Dendrion that moved into Morris Plains. Morris Plains is where that facility is. So we have 3 facilities in the country. We took the Morris Plains ones. Yeah.
Yeah. Good. Thank you.
You're welcome. Sorry to skip over that, but it's like that's not my area too. So I'm like, okay, let's look at. I just want to give an idea on the drug development process as to how it becomes complex as we go from one stage to the other. Okay, so phase 2 and phase 3 are very important because your process needs to be in a good situation. Now, normally, ideally, if you're in a biologics, by the start of phase 3, you need your final process, so no more changes. But because I said cell and gene, you're developing and running at the same time, it's very hard to fix your process at the start of phase 2. And a lot of times phase 2 is your pivotal process. Because in phase 1, you have, double-digit number of patients. So how can you set a process based upon, that small number? So we are learning and we are growing. So yes, we want a pivotal process. So we what we do is we define our must-haves and a nice-to-haves at the start of the pivotal. Without the must-haves, you cannot start a pivotal. But with the nice-to-haves, what we can do is, can we bring in those changes as the pivotal trial is ongoing, such that we can be ready for our commercial process. So what's happening in the phase 2, phase 3 versus the BLA and launch? You're really now looking at a larger patient population and saying that my current manufacturing process will work on a variety of patients. Now, this is not a given. A lot of times, this is when you find out where does this process fail, type of patient population cannot make the dose. That's also, we need to out in this, in a commercial setting, what to exclude, what not to exclude. So as we are progressing from the to pivotal, which is either phase 2 or phase 3, and getting BLA and launch, bringing all the knowledge that we've had from the preclinical phase 1, phase 2, about what should be that process that should for commercial, what should be our process development strategy, control strategy, what are our critical raw materials. All these have to be defined in the phase 2 and phase 3, such that when you're in commercial, it becomes easier and you don't have any hiccups. Now, assuming that you've been successful in phase 3 and now you're moving into BLA and launch and commercialization, some other things that you need to keep in mind are critical raw materials, but also backup vendors. A lot of the processes right now are dependent on single supplier. This is a huge risk for the whole industry, but this is again the reason, because the supporting industry has not grown with the cell and gene therapy area. You don't have a second supplier for certain critical raw materials. So figuring out who that backup vendor could be is very important. And then there's an element you see in all these, it's called comparability. As you move from preclinical to phase 1 to phase 2, phase 2 to phase 3, and then to commercialization, showing that the process and the product is comparable is extremely critical. And again, comparability can be a 2-hour talk, so I can't go more than that. I've explained some of the abbreviations here. IND is investigational new drug, BLA is biologics license application, and CQAs are critical quality attributes. The next slide gives a pictorial presentation of how complexity increases as you're going from the early to mid to late to BLA launch. This is again the preclinical phase 1, phase 2 / pivotal, and then BLA launch and commercial. So usually when you're in the early phase, these are small teams, one manufacturing site, definitely, a few apheresis sites maybe, and a couple of regions. So this is your early phase 1 world or universe, so to speak. Now, as you're going into phase 2 and phase 3, you're talking about larger teams, definitely multiple centers for apheresis. So you want to make sure your network is now grown in not only in US, but in Europe, in Japan. So you're increasing the apheresis collection sites. This will then give you a regional variation, not only in the patient population, but also in the way the sites do apheresis. So, and certain times when you have manufacturing failures, you can actually hone it into a particular site. So you know that they are not doing the procedures as they were required to do, or something is different because of which our batches are not meeting our required specs. As you are in pivotal manufacturing, you could have more than one manufacturing site so that you are now setting yourself for commercial success. And you could be working in 2 or 3 geographical regions. And when you're going into the BLA, now you're going like saying, okay, phase 3 is looking good, guys. So we have to file the BLA, which means now you're thinking about the global footprint. How many markets are we supplying with this? Multi-center, multi-manufacturing, multiple countries and multiple regions. And then now we are in commercial, and then obviously commercial, you want to supply the whole world. So you look at different strategies on how to supply the entire world. So why did I show this? I wanted to show this to show you the increasing complexity as you go from phase 1 or early programs into late programs. And at each of these steps, the transfers are extremely critical, because if you don't transfer it properly, then the next team does not know the mistakes the earlier team made. The next team doesn't learn from the history of the earlier team, and then they may be repeating the same mistakes over again. So transfers are critical. And over here, from my perspective, I can say CMC teams play a very critical role in the success of this program. So I think from here on, my talk is more on the CMC program management and product management. So why don't I pause here for a minute to see what questions people will have? Because I've given a lot of overview right now, and I think it will be helpful to pause for a minute.
Yes, Supriya ma'am is there. So ma'am, you can ask the question.
So Seshu, excellent presentation.
Yeah.
I just wanted to ask you, it appears a very complex process. And
Yeah.
for a clinical trial, how many patients are you looking at for a phase 2 or a phase 3?
So it depends. So very good question, Priya. So the thing is, it depends upon the indication. It
Okay.
depends on the population that we will ultimately serve. It depends upon the line of treatment. And then ultimately, whether are we going for a randomized trial or not. So all these things will impact on the size of the patient population. But it could be anywhere 100, 200, 300, 400, something to that tune.
Okay.
And the larger numbers are more for randomized trials.
Yeah, I know. Phase 3 normally is in thousands, right? Up to phase 2, you can do in hundreds. Yeah.
However,
And
cell and gene therapy, so far, we don't have a trial that's in thousands. So
oh, okay. Yeah,
this,
that's what I was wondering about,
yeah,
because the patient population and the target population that you're looking at,
yeah,
and especially the complexity of the whole process, the GMP side of it.
yeah,
Another question that I had, you said one patient is one batch.
yes.
How many cycles will the patient go through for the treatment? Is it one time, or do you have like chemotherapy, you go several cycles?
No, so that's why these are cures. It's
Mhm.
only one-time
Only
infusion.
one time. Okay.
So far, it has worked. Now, the remission rate or the success rate in the beginning in pediatric ALL is in the 80%. In DLBCL,
Excellent. Yeah.
it's in the 40%. So there's
Okay.
60% of the patients that may not. Another answer, so another way to answer your question is like, so randomized trial needs a lot more patients. So by
Yeah.
definition, it's a longer trial. So what the companies then do is they go to a later line, like a third or a fourth line, where you don't have to do a randomized trial. You do a single arm trial, again, 200, 300, or 100 based upon the trial. So you get approval in a third line.
Okay.
Then what you do is, so you get your first approval. Then we say, okay, now I want to move it into the second line. And here's my trial. Here's what I'm going to compare against. And this is my endpoints. This is how I will dictate that the trial is a success. That becomes a larger. But initially, companies want to get a quick approval in a later line in a single arm trial.
And what kind of endpoints do you define? Do you say 5-year survival? Because that's common for most of the oncology studies.
That's a good question. I don't think it's a 5-year survival. We get 9-month data, 12-month data.
Oh,
And,
yeah. Yeah, because it's such a new technology, right?
exactly.
Yeah.
And because each trial is different, right?
Yeah.
So each trial is, first you have to show that it's working. Then you have to show is it, what is it called? ORR, CR, PR, for how long, but it's not 5 years. That's
Okay.
for sure. Yeah.
Okay.
That's where the difference is between biologics and the cell and gene therapies.
Okay. Thanks.
Very good questions coming from a clinical trial person, right? So thank you.
Now, when
Yeah.
do you say your process is successful? I mean, just
Yeah.
because you cured one patient or two patients and
Yeah.
another two didn't cure, will you call it, or is
Yeah.
it an emergency approval?
No.
They have this word emergency approval.
So the trick here is how you define your endpoints in your clinical trial. It's very, very critical, right? So in the beginning, if it's a phase 1 trial, let's say, we'll say X number of patients. Usually it's less than 50, but in the 20 or something. And we will say that if it's 20 patients, let's say 10 or 15 have shown this much, 10 have shown this, and then we will consider phase 1 a success. Similarly, for phase 2, we will say if we have 100 patients, we need 80 to show this, 20 to show, 60 to show this, and whatever. And then we will say that it can go into the pivotal. It's not one or two. You definitely have three things. And Priya, you can help me out here. There is a, what is that? That term is not coming. But the clinical trial is designed by the MDs to say, a priori, we will say, what does success look like? And did the
Yeah,
trial...
you'll have to define your endpoints. What's your
Yeah.
primary endpoint? What are you
Yeah.
looking at? And then
Right.
there'll be some secondary endpoints as well.
Correct.
And you'll define it for particular population depending on the historic data
Yeah.
that you have available at that point.
And there's a statistical word that's missing me
Yes,
right now. The hazard ratio, right?
hazard ratio. Yeah, yeah.
Yeah. Yeah. So all those are, so there's a, 3, 4, 6 months discussion on how to design the trial. And there's another big story that I don't know I can share at this point in time, but seriously, the trial design is so important. It defines everything. And lots of times, the person who has designed the trial may not be there when the results are coming out.
Yeah,
And you're stuck
that
with
happens all the time. Yeah.
you are stuck with the design 5 years
Yeah.
ago, with a standard of care 5 years ago. Here today, it doesn't make sense. What do you do with the data?
Mhm.
So yeah, when I was at Lilly/Imclone, I actually recommended closing down a few, phase 2 trials because it didn't make sense.
Yeah.
Did that answer your question, sir?
Yeah.
Yeah.
And I just want to know,
Yes.
people have, bone marrow transplantation has been here so long.
Yeah.
What is the success rate generally?
I think it's pretty good. I don't have statistics, but that's the first. So even now, right, when the standard of care is stem cell transplant, HSCT, for even for a lot of the cell and gene therapy trials. And if that works, we will not give Carti.
Okay.
Yeah.
So it's a last resort. It's a last
Not
resort.
last yet. So here's the thing. Kymriah was approved in third line. Yescarta was approved in third line. Then, so the trials, we said, right, follicular, then, I was responsible for setting up in the manufacturing side about 8 different clinical trials for Kymriah. One of them was in second line DLBCL, which is called the Belinda trial. Similarly, Kite, Yescarta had their trial in the second line. Now, what happened is that this is public information, so I can share it. And this is why I said it's very important. Kite's second line data and trial is successful. Novartis's second line trial did not meet its endpoint. However, our drug works very well, even better than our competitors, and it's a safer drug. So this is where that design of that endpoint is so critical. What do you allow to the patient in terms of chemotherapy, in bridging therapy? How did you agree? What did we agree as to they can do while they're waiting for the drug becomes very critical. So this is a very recent example of how a perfectly good drug, but because the trial, for whatever reason, was designed in such a way that we did not meet the endpoint, we cannot move into second line. But Kite can move into second line. And it's not a last resort because there are people who are in 5th, 6th, 7th lines. We are in third line. And then we plan to move with our next generation platforms, we move into the second lines. And second line is pretty good because your first line is chemotherapy and maybe second line usually is the stem cell transplant.
Yeah, please carry on. Yeah.
So now so this is the whole overview section, folks. And now I'm going to talk about the CMC project management. I think we've covered an hour, 15 minutes, right? How long do you want me to go?
So you are muted, sir. Kanan, sir.
Yeah, sorry. You can talk for another 45 minutes, 10 o'clock.
No, 45 days. I'll talk for 15 more minutes and then we can
fine.
discuss.
Then we open it. Fair enough.
We can discuss. We can discuss. I can talk.
Yeah.
I can talk, no problem.
No, Seshu, because people are asking questions and you are replying. So that is part of our question and answer also is included in this.
Okay.
That's what
All right. Sure. So let me go back here. So now, folks, I have shown you the complexity in each of these phases. And there is a reason I showed all of this, because we have to pay attention to a lot of things such that we don't lose time and we don't waste time, right? So, and here's why I'm now going to talk about the CMC project management. What is CMC? CMC is chemistry, manufacturing, and controls. So basically, it takes care of all the manufacturing side of the program or the project. It includes research, it includes process, analytics, quality, manufacturing, reg CMC, release, everything, but it's all the manufacturing side of the clinical trial. Now, why is there a motivation to have CMC project management? Because it is a prevalent practice in mid to large companies. They have very organized, structured programs. But I would say that, even small companies need to have it. And we will say what size company and when. But what is the definition of a CMC PM? I say this is also known as technical project management. Novartis also says that. And it's a process that helps prioritize the tasks of a biotech organization. It puts timelines down on paper and helps visualize the goals and deliverables. So this way, it's a clear visualization for the teams that are working on these to say, here's my target, this is what I need to meet, and this is when I need to provide this to A, B, and C. Application of CMC project management, company, if it's a mid-size and beyond, you absolutely need CMC programs and PMs, which is at least greater than 3 molecules in the pipeline. If it's a small company with, let's say, only one molecule, you can define what the need of the company is, and then what form of project management do you need, and then take it from there. But certainly, if you have 3 or more, it's good to have a CMC PM or a program in the company. So what is it? The way I define it is it's an artery that runs through the organization. It helps channel information from various functional areas and ability to see the full picture from discovery through launch. We also provide regulatory insight for pipeline molecules. It can be used to help build a platform process, and it also provides a vantage point. It is strategic yet detailed oriented. And the way you can look at it is a one-stop shop. You can, the people who are working on this program can come to the CMC meeting, which usually can be weekly or every other week based upon the company. Reduces multiple meetings. You're all there at the table. You're discussing the program. You're discussing the issues. It's a common forum, and you can draw parallels from other programs that are working either in the same platform or the same program. So these are some of the high-level benefits of why one should have CMC project management. Okay, so why do we need them? They are an essential part of the organization. They will help connect the dots between various line functions. And in a minute, I'll show you what those various line functions are. They are a key to the success of the clinical programs. Now, in biologics, CMC program management is very important. But in cell and gene, CMC is everything. That's why I wrote it in capital letters. CMC is the backbone for the success of a cell and gene program, and it's in a very nascent stage, and very few people know how to do it well in the cell and gene therapy space. So that's why for the next revolution that's coming, CMC is everything. And therefore, CMC teams become vital to this rapidly moving field. Now, if you're a smaller company, the need is often overlooked because you have resource constraints and people constraints and time constraints. But it's good to be aware of what benefits this can bring, such that as you're growing in the company, you can make a recommendation. Now, this is from last year. According to this statement is from last year. I didn't update it, but according to the former FDA commissioner, the agency could approve between 10 and 20 cell and gene therapies a year through 2025, with as many as 800 such therapies moving through the pipelines right now. This is last year. I can assure you today's numbers would have moved much more than what this statement was from last year. So what does the CMC process, how, if you are in a company and you're thinking about CMC process, how would you start the setup? So first you identify the need. As I said, 3 or more molecules in the pipeline, you should consider thinking about bringing in a CMC project manager. You make the proposal to your senior leadership and you get their buy-in. Once you've gotten the buy-in, you first form a charter. And the charter will talk about the team structure, what's in scope, what's not in scope. That's very important to define up front. Then you figure out who's the governing body, add the governance, and how often would you go to the governing body, what is their scope and what's their roles. Then clearly define the roles and responsibilities of the team members, of the governing body, and of the boundaries. And then you go and set. So all this will take some mental mapping to figure out how do you want to proceed. Then you set up the teams, and then you chart out the objectives for the team, and you can start a project plan if you want, or figure out based upon the team how you want to go forward. And who are the members of the CMC team? So obviously, a CMC team will be led by a CMC team leader. And this leader, he or she will lead the CMC strategy. They will own the deliverables of the program, meaning they will own the deliverables of the manufacturing organization to the rest of the organization. And they should be the single point of contact. So some of these elements, I am highlighting them because if they are not done, then it causes a lot of confusion and a lot of unnecessary back and forth, which can be avoided if we kind of follow these ground rules in the beginning itself. What is the scope of the CMC team? Clinical trials, regulatory filings, process changes, comparability studies, tech transfer, change management, and everything under the sun. Like as a CMC leader, every morning when you wake up, you don't know how your day is going to end because there'll be a fire from one end or the other. A typical CMC team will comprise of people from the following areas: research, process, analytical, formulation, apheresis operations, manufacturing science, manufacturing, QA, quality assurance, QC, quality compliance, quality control, regulatory CMC, tech transfer, clinical supply. So this is a typical makeup, obviously, based upon the company, based upon the program. You may have other people that are coming in. They can be ad hoc members. And even from this, you'll have 5 or 6 as your core members, and the others can be ad hoc members. And it's good to define those in the beginning itself. Now, this is the composition of the CMC team. Now, how does this look in comparison to the rest of the organization? So the next slide talks about the global program team. Global program team has the responsibility of moving the program through the phase 2 into BLA or pivotal into BLA or phase 1 into phase 2. So this is the global program team's responsibility. They'll have a core team and they'll have an extended team. CMC is always a member of the core global program team. This global program team will be run by a global program head, GPH, and a global program director and a manager, so GPD and a GPM. At least this is how it's done in Novartis. So these are the main people. And then the core team members will include the CMC. PM, they'll include clinical development. These are the folks that will design the trial. Pharmacology, precision medicine, medical affairs, clinical operations. These are the people that will conduct the trials, meaning reaching out to the sites, figuring out when the patient is coming, when the manufacturing needs to happen, et cetera, et cetera. Then you've got global regulatory. These are the people that will file the IND and the BLA and any communication with the agencies. global marketing, Reg CMC. Reg CMC is a member of CMC also. Biostats, patient access, preclinical safety, patient safety. So these are the core members. Now, who are the extended members? Legal, research, biomarker, quality, medical, country medical people, and country commercial people, and health outcomes. So the way it works is the core team meets every 2 weeks. Extended team comes in once a month or once a quarter based upon which phase of the where exactly the team is headed and who is needed. Now, what can a CMC PM do in this kind of a setup? So is this. Our role is to harmonize between all the teams such that we can lead to a successful execution. So you've got the GPT here, which I just described. We have the CMC, which is our world right now, but we also have these sub teams. And as a CMC team leader, it's the responsibility of the leader to not only be very making sure that the GPT is aware of everything that the CMC is going through, but also to connect with the sub teams to see is there anything they need from the CMC group that they are not getting, or is there anything that CMC group needs from them, and then how to expedite. And each one of the sentences that I'm saying has examples, but obviously I can't go through that. But there's a lot of interaction that needs to happen between these sub teams such that the program is moving at the pace that has been defined. So
there are quite a few questions. I
Okay.
think Supriya and Anupati has a few questions to ask.
All
Supriya?
right.
Yeah.
Let's go. Go ahead.
Please go ahead.
Sorry, Seshu, I just wanted to say that I would like to leave. Excellent presentation.
Yeah.
I'll watch the recording for the rest of the stuff.
Thank you,
And
Supriya. Yeah. Thanks
That's
so
all.
much.
you.
Yeah.
Thank you.
Thank
Take
you,
care.
Supriya. Yeah.
Yeah.
Lovely. Anupati, what do you want to ask?
Yeah.
Yeah. As I understand, please correct me if I'm wrong. So the role of CMC is only during the early phase of the drug development
No.
Till the
So
end.
till the end.
CMC goes, it actually in the BLA preparation, they have that their role keeps on increasing. In early, maybe a little smaller, mid, even more, BLA, a huge portion. So the CMC is there all throughout. So let me take you back a couple of slides. Yeah, I think that will help you.
I got a bit confused.
Okay, so here, right? So in the drug development process, what's happening is you have a phase 1 trial. CMC team leader is taking care of the CMC portion for the phase 1. What does that mean? In the IND, there is a module 3, which is a CMC section, where you are saying, hey, this is my process. This is how I'm going to manufacture. This is how I'm going to release. All this is part of the manufacturing section. Now, phase 1 goes to phase 2, which means now the manufacturing process needs to transfer to phase 2. The process changes have to transfer to phase 2. We have to figure out that the tech transfer is happening in a proper way. The comparability we need to make sure that the process that's entering phase 2 is comparable to the process that was in phase 1. And also the IND filings in phase 1, then for phase 2, they're all interconnected. So the CMC folks are managing the same things with increasing complexity. Now we move to phase 3. So let's take a pause here because this is important. Phase 3 and also BLA, getting ready for BLA and launch. What happens when you're ready to file a BLA is you already now have your pivotal process. You have different sites where you have to transfer this process to and show that the sites, what they manufacture, that process is comparable to the current process. So that's where comparability come into place. We have to characterize the process because the process characterization will go into process validation. So you see here, a big prerequisite for process and method validation is that you need to understand what is your process looking like, what are the CQAs of the process, and then how do we set up the validation strategy. All this is the responsibility of a CMC PM. Then you have to develop process development strategy, process control strategy. What are the critical raw materials? What are the source documents? These source documents are provided to Reg CMC such that Reg CMC can do their BLA filing. So when I say CMC PM is responsible, they are not writing all of this, but they're members in their team that are doing all of this. So as we go from phase 1 to BLA, the size of your CMC team becomes bigger and bigger and bigger because the information that's needed to be provided to the regulatory agencies is larger and larger and larger. A simple example, the label on a drug. Yeah. I cannot tell you how many months it takes to fix the label, to get the artwork done, to get everything agreed and finalized, then to get translated to different countries. Just a label alone, I have a presentation that how many processes you need to set up and how complex that is. So CMC is part of everything. That's why I said in cell and gene, CMC is everything. I'm going to pause. Does that help answer your question?
Yeah, thank you so much. And then now I understood what role does CMC play, and then please come to GPT and can you please elaborate more on that?
Good, good, good. Yeah. This one or the next one?
The previous one.
Okay.
This one.
Yeah.
So now I understood, okay, the role of CMC. Now could you
Yeah.
just explain once again about the role of GPT now, because
Yeah.
now I have a reference what CMC does. So about
Good.
GPT,
Okay. So
just
so in a high level, GPT is the team that's making sure that the program is progressing from phase 2 or phase 1 into pivotal. Pivotal means pivotal, registrational, phase 3. They're all similar. Basically, what that means is the data that's coming out of these trials, we can use it to register the drug. Yeah. So to register the drug, it's not just manufacturing, right? You need to show it's safe. You need to show it's efficacious. You need to show that the PKPD is working. You need to show that within the patient, the cells are proliferating as we expected them. The statisticians have to look at it from what we discussed earlier, the hazard ratio, the endpoints, the statistics, the curves that are coming out, the Kaplan-Meier test. So all these things have to meet the required criteria. Manufacturing is only one little piece of that. There are commercial groups that are going to be looking at, can we get reimbursement from this drug in different countries, in different regions? If we don't get reimbursement, then how do we go about, right? So these are all the different functions that people are doing, and the global program team is bringing everyone together. But I'm going to just talk about maybe two for now, in addition to what I just mentioned. So clinical development are the MDs that are designing the trial. And clinical operations, this is in Novartis, are the people that are running the trial. So my interaction with these two is extremely close, and here's the reason why. The clinical operations are going to say, Seshu, a patient is going to come tomorrow. Can you manufacture? And I'm going to tell them no, because you didn't tell me this patient was coming today. I need to do A, B, and C. Or what I'm asking them is 6 months from now, can you tell me how many patients are going to come onto the site? Because I need to make sure there's enough people available. From a clinical development perspective, the MD would want to give a very high dose, or they would want to give a dose that's split into 3. And then I am the one that has to, meaning I, because I did that, but other people will have to tell the MD, sorry, I cannot make that high of a dose because my process does not allow me to go that far. Or I cannot split the dose the way you are asking. I can probably split it this way. So we are talking very closely with each other. They are my long-term friends now, because they'll say, yeah, I want to do this, but Seshu is going to say that this is not possible. Yeah. And the GPH will say, oh, but Seshu is going. I mean, they now talk CMC so well. It's very satisfying that they understand the risks and the difficulties of CMC. Similarly, the clinical operations from a capacity perspective, what is the capacity that our manufacturing site can offer them? Or if you are working with a CMO, as I said, CMO usually has 3 slots a month or 10 slots a month, let's say. So the clinical operations guy know that I can only bring in X number of patients. Beyond that, I cannot bring in patients. Yeah. Biostatist. So all of these people have their own functions to deliver to the global program team. And I'm just focusing on what we have to deliver from a manufacturing side.
Ma'am, there is a question from Ritika.
Yeah.
So kindly go ahead, Ritika.
Yeah, hi. I have one question. So, are these teams typically there in all companies? Is it like a requirement to have a CMC team and a GPT
Yeah.
team? And
Yeah, so this all this function falls under development. So it took me a while to understand why do they call this development. So this is the development organization of a company. So what that means is they are developing your drug, they're moving it through the phase 2 and phase 3 so that it can be filed. So they will do the BLA and then give it to commercial. Sorry, this jumps on me. So yes, they may be called differently, but global program teams are there for a particular therapeutic area or for a particular program. It depends upon each company how they want to do it. Is it one GPT for a therapeutic area, which is not easy to manage? So it's probably each molecule will have one GPT minimum. Now, when I talk about Kimraya, we had two GPTs, one for pediatric ALL, one for DLBCL. Now we are in the next generation platform. We started with one GPT, but it's a platform. So now we are breaking it up. We'll have multiple GPTs for the multiple programs that are coming. So yeah, it is a requirement because without that, you cannot file the BLA.
understood. Thanks. And I also have one more
Yeah,
question.
go
So,
ahead.
if we are comparing this process of CGT with biologics, so if
Yeah,
you can just tell me some high level differences, because
yeah,
how different is biologics when it comes to regulations, and how
yeah.
different is CGT?
I think there in the way I look at it is I was a biologics person before I moved to cell and gene in 2014. Yeah. So I brought all my biologics knowledge. So ultimately, it's very similar. If you know biologics, you can easily translate that knowledge into cell and gene because you still need, where did I say? Remember one slide where I said safety, efficacy, potency, quality, all that still remains same whether it's small molecule, large molecule, or cell and gene therapy. So the basics and the foundation remain the same. I think the biggest difference is in the characterization of the molecules. Flow is a technique and an assay platform that's used for cell and gene, whereas in biologics, we never use flow as much. And there's a lot of development that has to happen in these analytical assays. They're not mature. So they're very nascent. So we don't have the right kind of potency assay. We don't have the right kind of ELISA assay. We may want to do something, but it's not there. There are certain assays in academic centers, but they are not commercializable. They are not, we cannot use them in GMP environments because there's a lot of variability. You cannot qualify, you cannot validate. So these are all the problems that are there. But in general, if you have filed a biologic or if you have worked on an IND in a biologic and a phase 2, phase 3, you can translate that knowledge very easily into cell and gene.
Understood. Thank you so
Yeah,
much.
there's a workshop I used to run for five. Well, I was teaching the CMC workshop as part of a larger workshop. It's called the Drug Development Boot Camp. So for 5 years there, I was talking about the biologic CMC. Pretty much the issues are similar. It's just that some problems may be different. So here, as for example, the batch to batch variability is a huge thing because as it is, the process is variable. You have less number of ends in the beginning to standardize the process. And so you need to figure out where the variability is coming from in the current process. Now you're adding this donor to donor variability. So each batch is going to behave in a way that you cannot predict. So then from your experience, you have to define a process with wide enough range, but narrow enough that it can be released. So those are the kinds of challenges. So the control strategy for the manufacturing, for process, for analytics is much more challenging in cell and gene when compared to biologics.
Seshu, when I
Yeah.
look at your core team, don't
Yes.
you think a hospital will be the right environment to do the whole process with a, R&D there or something like that?
So hospitals, so GMP manufacturing is very, very difficult. You need
Mm.
a whole organization that has the mindset of GMP, that has done GMP. Hospitals will say they do, but they can't be bothered with the kind of regulations we have to follow, the procedures that we have to follow, the burden that is there to maintain a facility in an inspection ready mode. It's way too cumbersome, number one. Number two, hospitals will not be able to do the autologous cell therapy manufacturing, which is 9 days and stuff like that. Hospitals cannot do. What they can do, and where the industry is moving towards, is how do you automate the whole Carti to make it so simplistic that it can be done in one equipment and one whatever, and that can then be done at a hospital site. However, this is another 2 hour topic, sir.
No, I agree. No, I just wanted to get a
No, no, no.
general
I'm
feel.
just saying because once you come to that model, which is called point of care manufacturing,
Ah,
then you figure out who owns the IP. Who is the drug? Is it a Novartis drug or is it a hospital drug? Because if it's a Novartis drug, then the quality requirements are very high. If it's a hospital drug, how do you know? How do you troubleshoot if a manufacturing process fails, right? If you don't have these strict requirements in the beginning, then tomorrow when you cannot succeed in a manufacturing, you don't know where to trace back. Hospitals will not have the backup vendors, right? Having dual vendors, having this, having that. They won't have any of that. If a vendor has a problem, they don't have the number of people to troubleshoot to say, oh, these bags are not meeting our quality standards. What do we do? Do we stop manufacturing or do we give a substandard product to the patient? So hospitals are still far away.
okay. Because in India, I remember a
Yeah.
lot of people started doing this stem cell therapy illegally. They were not approved. And we used to have a lot of patients coming from other countries because those countries it is not allowed. But
Yeah.
they said, no, we want ultimately my patient should survive. We don't
Yeah.
mind taking the risk. Anyway, in America, they will not survive. So why not I
Okay.
come here?
Yeah. So, and there are a lot of hospitals in Spain and everywhere that are doing that. They are administering the point of care the drugs to the patients because Carti they're able to do. The issue will come in the consistency and the standardization. So
Agree.
if that particular doctor can do whatever he wants to, but if he has to transfer it to another site, does he have the procedures to transfer it to the other site? Will it work the same way? Can he show they're comparable? So all those questions become very challenging.
Because I suddenly find there a lot of interest in doing CAR T cell in India. But
Yeah,
I don't know whether we are yet ready for such
yeah,
a CMC program. Personally, I
right.
mean,
Correct. So see, that's why I said if it's a smaller company, which usually that's how they start off, you may want to figure out what kind of management you need, right? Something you need to standardize.
mm,
Do you want it to be CMC PM or something else? That's up to the discretion of the organization. But I'm just trying to show the structure, what it'll bring in terms of benefits.
okay, yeah, understandable. Yeah,
Yeah.
please go ahead. Yeah.
Okay. Oh, sorry. Was that Ritika? Did this explain the GPT to you?
Yes, yes, completely.
Yeah.
Thank you so much. Yeah.
Uh-huh. Okay. So I talked about this. Yeah. One thing to remember on this slide is that CMC team leader, right? How can he or she be successful? Interacting with sub teams on a continuous basis, engaging the team members, providing timely updates. Why is this here? Lot of times, CMC is always on critical path. So GPT wants to move at a speed of light. CMC will say, sorry, you can do what you want to, but this is what I can do. This is what I tell the folks saying, you can plan whatever you want to. Here's my plan. This is what I can deliver. And there's a 50% probability of success that I can deliver because there's so many other risks. So, how do we provide timely updates and to whom is very important? If there's a deviation, my philosophy is right away go tell them there's a failure, there's a deviation. I need 6 weeks, I need 4 weeks, I need whatever. I've seen other groups that take 4 weeks to come up saying there was a failure 4 weeks ago. So, how do you provide those timely updates to your customers such that they can have trust in you? That's the most important thing. If things are going well, you need to let them know. But if things are not going well, you need to figure out how to inform them. At the same time, you need to Provide a solution. You cannot just tell them it's not working, because then GPT will come back with 100 questions and you will not have a zero, you'll have zero answers. So you need to figure the problem. You need to say, okay, what can I do to mitigate the risk? What's the impact to timelines? What happens? What's my plan A? What's my plan B? And then I go to GPT. But again, I can't take a month to go to GPT. I should go within a week of when a problem has arisen. This is how you would manage a risk. And this is how you would communicate actively to say, this is what is happening, folks. And then if all else fails, you seek help with technical challenges and say, I cannot solve this, but this is what I'm doing. I just want you to be aware. The other big thing that the CMC has to do is to partner with your regulatory CMC folks and quality folks. This is very, very important. Now, if you've worked in a GMP environment, usually the SMEs and the quality folks and SMEs and Reg CMC folks, there's a tension. And the tension is because each one is coming from their angle and they want to impose certain things on your manufacturing. But the idea is how to create harmony between these three, listening to each other such that the subject matter expert is saying what is feasible, what is not. Quality is saying what is acceptable to them versus not. And Reg CMC is saying what is acceptable versus not. So this is how you create a good environment in the beginning such that you set up yourself for success. And so that's, again, the proactive discussions and brainstorming helps in all of that. I think there was a question. Was there a question? No. Okay. So these proactive discussions are very important, folks, because then what happens is, as the process folks and the analytics folks are setting up the process, as a CMC team leader, you know what is the expectation of the quality folks and the Reg CMC folks, meaning what kind of documentation that Reg CMC will need for IND, and am I generating data that will go into that source document or not? If I'm not generating data today, then I'm wasting time, and I need to then later on come up and generate that data. So these things help in saving time. Similarly, from a quality perspective, especially CGMP quality, there are certain areas where they cannot budge. There's no flexibility, and we need to make sure that we are aware of those such that our program can address those as we move forward. Then, okay. So I think the next three slides are, talking points, the points that we want to consider and why. And as I explained, the drug development process is long and complex. When I say long, what are we talking about? It could be 5 years to 8 years before the drug can be launched. So if you imagine a mistake that was made in the phase 1 or preclinical, if it's not rectified as we go and it becomes a big issue at the time of BLA, we have lost valuable time where we could have rectified that error, So that's why it's long. The more we set the right foot forward the first time, better for us. And obviously, it's complex in addition to being long. So the more things we simplify, the better it is. Now, in order to be competitive, it is prudent to be aligned across the program. Mistakes can prove to be quite costly. So therefore, anytime you have an issue, going to the GPT to say, folks, this is what I'm seeing in the CMC area. And this could be a residual that acted differently, a spec that acted differently. How does that change the label that the GPT is going to ask for at the BLA? So having that alignment and discussion and brainstorming is very important. So that brainstorming means crosstalk, early involvement of folks, and sharing of lessons learned across the program, meaning you could have multiple programs. So the phase 1 teams can talk to each other. The phase 2 team from one program talk to the phase 1 so that they can provide what difficulties they faced with the FDA or the EMA interactions. The launch folks can tell, this did not work for us, this label, this gave us a problem, such that now when I'm in a phase 1 program, I rectify all of that from the beginning. So this way you have learned from the mistakes of the past. So this is where I feel CMC team leaders are very key to ensure this cross-functional dialogue and a smooth transition between phases. And developing proper risk mitigation strategies and technical justifications to overcome the risks are very important. Now, why do I say that? There is risk in everything you do, yeah? Question is, how do you mitigate the risk? And do you have sound technical justifications to stand behind that? So I'll give you an example here. My validation for the BLA, SBLA for Kymriah for DLBCL, we had so many deviations in the last 2 months, 3 months, that there was no way we could have filed on time. So we came up with a good strategy, the only strategy that we could do to meet the timelines. So my management asked, what is the POS? And I said 50%. And they're like, do we have any options? I said, if you want to file on time, there is no other option, but I'm not going to file this if I don't have your buy-in and backup. So they all agreed, saying this is what we will do. So how do I mitigate the risk, right? So we file it, but we're already working to say that if an agency objects to this, this is how I'm going to mitigate it. But luckily, with the 50% POFS, my validation strategy was approved in EMA. It was approved by the FDA, actually loved it, and they didn't ask for further questions. But I was ready for technical justifications such that if they push back, we were ready with the answers. So having these things, you can do miracles in your program. And then this helps in keeping the program viable and competitive, because now you're able to meet the timeline, you're successful, and you don't lose any additional time. What are the benefits in this slide? What does success look like in the next one, and something else in the third one? So what are the benefits of having the CMC program management? So obviously there's cost saving, there's time saving. You're looking across the organization and aligning on strategic objectives. So you're not only guiding your own team to work properly, but they are aligned with the company's objectives, which is the most important thing. If you have a proper CMC program management or project management, you're removing the silos across the organization. So, what you will see in large organizations, and I have seen this and you will see it too, is that the process group doesn't talk to analytical group. Within the process group, one group may not talk to the other group. Quality, manufacturing are not talking to manufacturing science and technology. So, or manufacturing science and technology is talking, but other groups are not talking. So when everybody comes to the table and you're addressing an issue, you're actually removing the silos, you're making crosstalk happen, and you're ensuring that the program is moving forward. It's very, very important. There was a time when I was pulled into the Kymriah validation from my stem cell program. I had to come from the second floor to the first floor to make people talk who were next to each other. I'm kidding you not. I'm like, I'm coming from the second floor so that you two can talk to each other and you're right next to each other. So this is how you remove the silos. Yeah. Integration with new SCM models is critical for personalized medicine. Mapping interdependencies of projects across the platform. So again, the interdependencies are very important. If process is doing something that could change something, as a CMC team leader, you need to know, because you need to understand what are the implications to a timeline, what are the implications to a tech transfer, to a comparability, whatever that is. And so these interdependencies are the sole responsibility. The CMC team leader should look into all of this. Targeted focus and actionable alignment with senior leadership team. What does this mean? Very quickly escalating issues if you see an issue. If not, you can say things are going fine. But then getting their buy-in is very important, because you're not alone and the risk has to be shared with everyone. As you're doing all of this, you're going to identify gaps in either procedures or whatever, and then you're making sure that you're overcoming that. I've mentioned this before, a good hands-off, handoffs from phase 1 to phase 2 to BLA, and then end-to-end process improvements and driving lean initiatives. So all of these are the benefits of CMC, having a good CMC team leader. Examples, okay, so this is the benefits. So now let's look at what happens when there is lack of alignment. So if there's lack of alignment, you'll have improper stakeholder management. There'll be conflicting messages received by different organizations. This has happened, guys. Change gets approved by a health authority, but there is no proper implementation plan. Manufacturing facility is not ready for your first patient, first manufacture. There could be delays to manufacturing delays could impact the clinical trial completion timelines. There could be delays to tech transfer because new site readiness is impacted. Or even worse, the improper execution of comparability protocol can actually kill a program. And overall, there could be delays to timelines. Now, there could be delays to timeline even if everything was done properly. Don't get me wrong. But if you didn't do some of these things properly, you definitely will have a delay to the timeline. And on the other hand, what does success look like? Good stakeholder management, program meets its timelines in a reasonable manner, risks and delays are communicated in a timely manner, CMC teams are high performing, teams speak with one voice, GPT teams and governance boards are engaged appropriately, trial meets their endpoints, BLA MA is filed per target, and program launches as expected. There is one more slide, but I'm going to pause here because I spoke a lot right now. Questions, guys?
Yeah. Now, basically, when I look at all this,
Yeah.
you started as an MSc in biotechnology so many years ago.
Yeah.
Today, if we were to, define a curriculum in biotechnology, because these processes are very important now.
Yeah.
So, what emphasis will you give? And will you give how many
Yeah.
credit course for this?
So that I don't know about credit course, but I think having an introduction to regulatory filings would be a good one for people who would be doing biotech. Having an industrial viewpoint and talking about these control strategies or validation strategies or qualification for assays, those things are very important. So people coming in knowing what challenges can be there would be very helpful, I would say. And, I mean, I'm just in this is 60-second thought in terms of curriculum, but that's would be what I would say would certainly be recommended.
Yeah, because when we were teaching you, people we teach you what is hybridoma technology, genetic engineering. But today, when I look at it, you are going to give a product to a patient.
Yeah.
So the quality control and regulatory are more important than even the process itself. The actual process is probably 5%, but the rest are consuming 80-85%. Am I right?
I'm laughing because I think you can take my workshop and convert that into an educational course. Seriously.
No,
It touches on all these topics.
Yes. No, why I'm saying is, when because I'm involved in academic programs over the years, I always feel that, we must change the way they come. Yes.
Yeah, it has to be relevant.
Otherwise, student says, what I learned is of no use to me when I go to an industry.
So I think that's a good point. What we can do is combine, so add some things, but combine what was taught over 4 semesters into 1 or 2 semesters to say, okay, you don't need to know exactly which promoter to use in which gene, but you need to understand, you need to pick the right promoter, because as the indication changes, the promoter is going to change. As the theory behind this changes, the things are going to change. Do we need a co-stimulatory domain and stuff? So as long as people have the ability to synthesize this information together, they don't need to go deep. So it has to be broad, but sufficiently deep, but not too deep.
Yeah, because when they go for a job, people would like to know, are you familiar with these terms or have you heard of it? Because nobody wants to know.
Exactly. So having this regulatory overview is very important. Very important. Yeah. That can be a degree in itself.
Exactly. I could see that. I mean, there is specialization in that area.
Yeah, exactly.
Earlier, we did plant tissue culture, animal tissue culture, like that. Now, I think regulatory specialization is also equally important.
Yeah. Quality, regulatory, and something process, something like that. Those could be very helpful.
Very true. Yeah. Please go ahead.
No, so I have only one more slide, which talks about the tech transfer. But I think it's a good time to pause so we can, if people have questions, I'm happy to take. If not, then see, this is the tech transfer slide that I have. That's about it. And that was just as an example of a case study, if people need it.
Yeah, actually, you can quickly go through it so that people know what is the technology transfer strategy. Yeah.
All right, so I came out of the slideshow. Let me know if the slideshow works. Does it work?
Yeah, it is working.
Okay.
Yes.
Okay, so I have emphasized tech transfer so much because tech transfer is a very integral part of this whole process. So what are we doing? We are taking an initial process from a lab. We are taking it from a lab to a manufacturing facility. From a manufacturing facility, we are then taking it to a second manufacturing facility because now we need more of the same drug. Then now we need a European or a regional manufacturing. So we now transfer the same one to another facility. And in all this, while we are transferring, we're also trying to see if we can do process improvements. And as we are moving from phase 2 to phase 3 to BLA, the requirements on our process become more. And whatever we've learned in phase 1 and phase 2, we want to include that into our process going forward for launch. So we have to make those process improvements as we go. So therefore, tech transfer plays a role in all of this. And comparability by the same topic, tech transfer, you do the transfer, you have to show it's comparable. So they both go hand in hand. So what is tech transfer? It's a systematic integrated process for transferring knowledge from one site to another, which consists of information transfer and demonstration of the transfer success. So there are different phases: evaluation and decision, planning, preparation, execution, and post-transfer assessment. So in the initial evaluation and decision, usually there is a make or buy. What does this mean? Are we making this drug internally in our site, or are we going to CMO? Yeah. Then we are looking at allocation. Sorry, one second. So generate the allocation proposal, secure leadership and endorsement. So this is the first step. As you're working in bigger companies, you need to get the management buy-in as you're moving through. That's why this leadership endorsement becomes very important. Then the planning phase is when you are nominating your tech transfer team, both from the sending side and the receiving side, figuring out what is the high level schedule, when, what are the limitations at each site, because of which the schedule will change based upon the limitations or the abilities. So is it going to take 6 months for the transfer, 9 months, or 12 months? Because that is going to impact your clinical trial timelines. And the length will also impact the budget. So we need to make sure there's at least some good planning in the beginning. And once we've planned that, then you secure the funds for approval, kick off the project, and prepare a tech transfer master plan. So this will lay out the high levels, milestones of how we are going to go through. Excuse me. And then you're preparing for this. This will depend from company to company. It will have its own preparatory phase, but this is what is how it's done at Novartis. So you open what is called a change request. Open a master change request, which is going to say that we're going to bring this molecule, this program into this facility. So that's the overall master change request. Below that will be different other change requests, but those will all refer to the master request. Assemble a technical TDP, technical development plan, which talks about the manufacturing and the analytics. The first thing is to do a gap assessment between the sending site and the receiving site, which will then tell us what are the areas we need to overcome in the receiving site. And then prepare detailed project plans and then prepare protocols. This is the third stage. Now we're coming to execution. So we are transferring the analytical methods, we are transferring the process, we are procuring the materials, you're doing the equipment, you're buying the equipment, you're installing it and qualifying it. Then you do technical runs, which are called as feasibility runs or some other name by different companies. Once you're shown success at technical runs, then you move on to your comparability runs. This is where you'll say, I'm going to you'll have a protocol that says, I'm going to do 3 runs. They're going to meet this criteria. And these comparability is like, it'll be comparable to the runs run in the sending site, yada, yada, yada. It's a, big one. And then as you're doing this, and if it's a later phase program, then you're talking about process validation. And then you're putting all this information and you're submitting it in the dossier, either it's an IND or a BLA. After you've done the transfer, you prepare and approve the tech transfer report, follow up whatever tasks are left behind, and do a perform lessons learned exercise. Very, very critical. The line functions that you want represented in your tech transfers are manufacturing, engineering, supply chain, sourcing, quality, safety, regulatory, analytical process, and finance. Finance is also important. Sub-team leaders will be attending these teams. They need to be proactive. They need to take care of the action items in a timely manner as defined in the tech transfer master plan. And the sub-team leaders either should attend the meetings regularly or provide a delegate. So this is also important, because a lot of times everybody has conflicting meetings and then they don't have a delegate, and then you miss out because the time is very precious. So this is just one sample of, how to do a successful tech transfer. I'll stop sharing for now.
Yeah.
So I know it's a lot that I presented there, but it's basically walking people through such that you get a flavor of
Yes.
where the challenges come. And challenges are several, like, on a daily basis, it's some fire somewhere or the other.
I think I must really compliment you, because for me it was a great learning experience that, when you say a product is launched, you don't know how much effort has gone behind it. Only
Yeah.
when we listen to a talk like this, we
Yeah.
suddenly realize that, look, how many people are involved and how many steps are involved. I think it was amazing presentation, and your experience showed
Exactly. This is,
that you have experienced
yeah,
everything.
I've done every bullet that's out there, every blue word that's out there. I have experienced it, right? So this is my life, so to speak.
I know.
And things go wrong at the worst time, to be honest, like really. So you have to be prepared with a plan B all the time.
Surya, you can ask your question.
Yes, sir. Yeah, thank you, sir. Thank you, ma'am, for the wonderful talk. I have few questions, but most of
Yeah.
them are related to Carti cells.
Yeah.
So first, considering that this Carti cells which have been made are CD19 specific, which
Yeah.
means that they are targeting B cell related conditions,
Mhm.
and considering that the relapse is very high in B cell related lymphomas as well as in other like B cell Hawkins lymphomas,
Mhm.
like what do you think on the down regulation of this CD19 molecules as well as the loss of antigen of CD19 and not sufficient CD19 being present during the relapse?
So these are, I mean, excellent question. This is where the field is looking into how to prevent all of this. So what is causing? So there's when the relapse happens, is it a CD19 negative relapse? Is it a CD19 positive relapse? Number one. If it is a CD19 positive relapse, if it's negative, we can't do anything because the CAR-T is designed for CD19. But if it's a CD19 positive relapse, then is the dose sufficient? Is the transduction efficiency, do we need to, what do we need to tune in or fine tune in order for it to be for it to not relapse so quickly? So an example I can give, both pediatric ALL and DLBCL, they have a dose range. And then in the beginning, so this is actually a very, very life learning example. Based upon the clinical trial, it was shown that any place in the dose range you can give, there is no impact, it will be efficacious, right? So very low dose also, the patient showed remission, high dose also they showed. So the manufacturing has the flexibility to go through the entire dose range. And that is how the manufacturing procedures were written, such that now, from a manufacturing perspective, when we freeze the cells, we need to make sure our potency is within limit, our this thing is within limit, all those things have to be within limit, and things can go out of control. So we were trying to make sure that we are within the range such that we can release the product. As we now a commercial drug, we have patients coming, no exclusion criteria, all patients can come in, you realize that you need to be on the higher end of the dose. But the manufacturing procedures have not yet caught up with the higher end of the dose. So it took 2 years. It took 2.5 years, half year to figure out the problem. From the time the problem was figured out, to see that it's not a regulatory filing, it's just a GMP SOP procedure modification, 2 years. And the person in my group worked on it, and he's such a calm guy, the calmest guy. Now he's lost all his patience just changing this one thing to say, how can we give the maximum dose possible, and how we change the manufacturing procedures. So to answer your question, I think there's a lot of groups that are looking at, is there an on-off switch on the CD19 such that it doesn't need to be on all the time, so it's killing the B cells all the time? So all these things are under investigation, and I think time is of the as we get more information, we can figure out how to augment the CD19. Certain, like I think the Yescarta medicine does have a better efficacy and a remission rate, probably because if I'm not mistaken, they're now as you also move into second line, you will see that it may work better because the patients are healthier, and therefore they may be able to receive and react better. So there's a few things that we have to look into to come up with that answer to the question that you asked.
Yes, I have my second question.
Yeah.
This is regarding, like, we have been doing TCR alpha beta depletion for haploidentical transplants using Clinimax plus machines. The
Yeah.
thing is, like, how instead of just isolating the T cells and changing and preparing the card, like, can we go one step on isolating the TCR alpha beta cell, like depleting the alpha beta cells and only concentrating the gamma delta, and then going for a CAR, or only a TCR therapy can still work? So how different those
Yeah,
tools are?
so that's a good question. I don't have the answer, but I know companies are looking into gamma delta T cells, companies are looking into TCRs, and companies are looking into TILs. So that's the best I can say without actually, that's a good way to look into it. I would not have the answer.
Okay, ma'am. My third question is, like,
Yeah. Oh,
with
sorry, one more
respect
thing.
to...
Surya, sorry, one more thing, right? Let's assume that may be an option. But right now, the drug that is approved has this way of, like, we take the apheresis, we select the cells out through CD3, CD28, and then we modify. So if in future the manufacturing process changes, even though it's the same indication, it will be a different drug. So if it's a different drug, you need to go through phase 1, phase 2, phase 3. So you're talking about 5 to 6 years. So just saying, this is
Yes, ma'am. Yeah, I get it with respect to the clinical trial aspect. Yes, ma'am.
Yeah.
And my
Yeah.
third question is, like, considering that Cartis are once administered in the patients, like after chemotherapy, and usually, like, they are so vulnerable for like, in terms of cytomegalovirus infections and other bacterial infections are very common. And then we are also introducing something called cytokine mediated storm in Carti
CRS.
cells, which are not there in normal peripheral blood stem cell administration, or a stem cell administration, where we are only trying to target or tackle GVHT. But in this case, we are actually going to be tackling 2-3 aspects other than GVHT, which is not going to be enough thing. But how in that case, what I feel is, like, whether the stem cell transplants being the standard care of treatment right now, are they still going to be the standard care, even considering the cytokine storm being a major issue in Carti administration?
Okay, so I think I've gotten the thread, but I want to digress for a minute. So the GVHD, right, stem cell transplant, and the GVHD that you mentioned, my first project at Novartis was the stem cell project. We were looking to just modify the cells, not do any gene therapy into it, such that GVHD is not there. And so that is a company now called Telaris. It used to be Regenerex. That's the phase 3 IND that I filed for Novartis, and that got approval in 30 days. So they do this such that in a kidney transplant, when you modify it based upon that procedure, the donor does not, the recipient does not reject the kidney. And there are examples of people that are now, they don't have to take any immunosuppressants. 10 years. It's been 10 years since the kidney transplant. They're off immunosuppressants. One person has even delivered a baby after having a kidney transplant and the Telaris or the Regenerex medication. So I just wanted to share that because you mentioned about the GVHD in stem cell transplant. Yeah. Okay. So your second question is the CRS. So the companies have done a lot of work in managing the CRS, either proactively or after giving. So each company has a different method in managing the CRS. Among all of them, Novartis drug is the safest. This much I know, because our grade 3, greater than grade 3 is very less, whereas the other people have either neuro affects quite a lot or CRS. So they're all learning on how to proactively decrease the CRS. And now they're moving towards steroids as a way to even reduce further. Tocilizumab is one that's been accepted along with Kymriah, and I think Yescarta may be doing the same. So CRS is now being managed. So your question is, knowing that CRS is an issue, would you consider stem cell as a standard of care or not? I think if the patient can get cured with stem cell transplant, that would be the first line. I don't think physicians are running to give their patients to Carti yet. So there is so they take the first or second or maybe 3 lines of treatment before they say we need to go to Carti. So this is something that we have experienced in the commercial world, so to speak.
Yes, my fourth question is, like, what is your take on closed system of producing Carti cells like Clinimax progeny in
Yes.
comparison with open system where the process probability of contamination is high in terms of endotoxin mycoplasma?
Wonderful, wonderful. So that is the system everybody is going towards. You want to close them. So what are the advantages of closing the system? If you have a closed system, then within the GMP environment, you've got a grade B and a grade C environment. Grade B environment means a lot of environmental modeling and double gowning and stuff like that. Grade C is a more open ballroom type environment. You can put your prodigy A, B, and C, and you can manufacture. It's closed, so you don't have to worry about sterility issues and stuff like that. That is the future. That's where things are moving. Unfortunately, for the Kymriah and the Yescarta and the BMS procedures that were the first time approvals, they all have a wave bioreactor wherein they are culturing. So it's a 9-day culture. Prodigy is not fit for doing those things. I think Prodigy has its own limitations, but the next generation platforms are using either a Prodigy from Miltenyi, Cocoon from Lonza, or something else, but that is the future.
Yes, ma'am. Thank you so much. Thank you, ma'am. Thank you for the answers.
I think Yatendra, why don't you wrap it up now? I think for Seshu must be quite tired now.
I can talk, sir. That's not a problem. If there are questions, I mean, this is CMC has been my bread and butter. I live it. I love it. So it's fine.
Yeah.
Ma'am, it was indeed a very comprehensive talk, and I think you have crystallized years of experience in like the drug development process within 2 hours.
Yeah.
And we are like extremely thankful to you. You accepted our invitation from KK Biotech,
Yes.
and really excited to hear you, and looking forward to be in contact with you for like further leads.
Yeah, in fact, she said she's willing to give some more, lectures on each one separately. Today, she gave us an overview of the whole thing.
Yeah. So, because there's a lot, and even the CMC, right? Just the CMC, even though it's only words, it really helps define the process so you can avoid some pitfalls as you go forward. And people who have done CMC, when they hear the talk, they come and say, they're like, my God, I've been doing CMC for 15 years. You're so on point on everything you're saying. I said, yeah, because, it's like, you want to avoid the mistakes that others have made, that you have made and stuff like that.
In fact, I wish we have CMC in education also, so our success rate increases.
Exactly. No, but I also want to thank all of you. I think this is great questions, folks. Hopefully you've learned something from my experience. Thank you for the opportunity. It was a very, very busy schedule for me, but I wanted to do it, so I did it. So it's good comprehensive view for everyone. Happy to come back again, so that's not a problem. So thank you.
Thank you, Seshu, for pulling some time out to present.
Yes. And 1:30 in the night, like this is what
Anyway,
all the best wherever you are going to be. And
sir, excuse me, sir.
Touch.
Like I think Mr. Sunil has a question because his
hand is up.
Please.
Maybe we can have a quick answer.
Okay, I'm so sorry. I'm keeping you awake for. I mean,
no. It's daytime for me. No.
It's a daytime for her.
Okay, 1:30. Okay, sorry.
Okay.
It was an amazing talk. So nice to listen from the horse's mouth. Who has done all this.
You.
Yeah. So my question is, I'm interested to know how are we placed right with the signaling molecules, like you showed the CD8. with the signaling molecule, right? So CD8 is especially for the to avoid this T cell exhaustion, I believe, and enhancing the signaling, right? So we have gone to the third generation, I believe. So what are the other signaling molecules that we have attached to the Carti antigen?
Okay, so that I will not be able to answer, number one. But number two, when we characterize our product, we look at the CD4, CD8 ratios, we look at batches that are successful. What's the ratio of CD4 or CD8? Is it better to have one over the other? And then within the T cells, what type of T cells would we want to do? So all these characterization assays, either they're in place or they need to be developed, but they help us in figuring out what should be that ideal product composition. From first, second, and third generation Carti, it was the addition of the stimulatory domain, right? So first it was one, then we added the next, then we added the third. That's when the molecule actually became active and then was able to go after the CD19s. But T cell engineering, that's like preclinical, and that's not my forte. Sorry.
Okay. Thank you so much. Thank you.
You're welcome. No, that's fine. I can look it up and see if I can share something on that.
Thanks.
So once again, I would again like to thank Dr. Seshu Tyagarajan for such an interesting talk and like enlightening with this latest technology of Carti.
Yeah.
And also the participants who actively participated in the session.
Yes,
so
definitely.
Thank you, ma'am, once again. And thank you, Kanan sir, for giving me this opportunity to moderate the session.
Thank you, guys. It was lovely meeting all of you. You're on mute, sir.
So you are muted, sir.
I think it is so nice to have listen to the students whom I had chance to know them at various phases and see how they are talking modern science. Nothing makes me more happy than that. Thank you so much, Seshu. Yeah,
you're very welcome.
I feel very happy. Yeah,
my pleasure. Yeah. Thanks a lot. Have a wonderful
day. Have a nice day. Have a nice day. Yeah. Bye-bye.
Okay. Great question.
Thank you, everyone.
Bye-bye. Bye.
Thank you.
Thank you. Bye.
Bye.